Aloha everybody. I will focus my presentation today on fatty acid catabolism, which is described in chapter 17 of your textbooks. I begin my presentation by reading Leviticus chapter 3 verses 14 to 17. And he shall offer thereof his offering, even an offering made by fire unto the Lord, the fat that covereth the inwards, and all the fat that is upon the inwards, and the 2 kidneys, and the fat that is upon them, which is by the flanks, and the caul above the liver, with the kidneys, it shall he take away. And the priest shall burn them upon the altar. It is the food of the offering made by fire for sweet savor. All the fat is the Lord's. It shall be a perpetual statute for your generations throughout all your dwellings, that ye eat neither fat nor blood. Then I normally ask you, how do those verses make you feel? And I usually get the answer that, oh, this is the Old Testament, doesn't really apply to us anymore. And then I would normally ask you, what do we give the Lord as an offering? And if you know your religion well, you should answer that we always offer the Lord the best. We give the Lord the best of everything. When the lamb is sacrificed to the Lord, it's supposed to be the firstborn lamb without any blemish. That lamb is being raised with the family, and then it's slaughtered as an offering. And do you think what is described here in this verse describes a bad thing or a good thing? Of all the things that we can give the Lord from a slaughtered animal, we give the best, the fat. Now, fat is not a very popular food group, or fatty acids are not very popular molecules. As a matter of fact, I rarely hear people praising that they're eating fat or being excited that they will be eating fat. Most people that I speak with usually are somewhat repelled by fat and don't want to talk about it. Protein is a very popular and kind of good food group. Carbs, bad. Fats, bad. Proteins, good. We'll see how you feel about that by the end of this course. You probably will feel the same, but you will have a little bit of a different view, hopefully. So, again, keep in mind, we're offering fat, which is the best, and we're offering that to the Lord. Now, fat as a molecule is the most efficient way to store energy. It's been determined that for every gram of fat, there are 9 calories of energy. These calories, though, by the way, this is a capital C, which means kilocalories. So if you're used to the calories that you have learned about in general chemistry or organic chemistry, food calories are kilocalories. So this is basically 9,000 calories of the regular ones, but 9 food calories. Glucose, 4 calories per gram of dry mass. Protein, 4 calories. Alcohol, 7 calories. If you remember, when we've talked about alcohol fermentation, I told you that this is the second most caloric substance that humans consume after fat. And you can see now that indeed that is the case. Surprisingly, carbs and protein carry the same amount of energy based on their dry mass. So, fat carries the most energy. Now, if you think about it, you probably don't know that yet, but why is that? Why is fat so caloric? Why so much energy? Think about the structure of fat. What is it made of? Primarily hydrocarbons. Glucose, it's carbohydrate. It has carbons and hydroxyl groups. Proteins also have a lot of nitrogen, hydroxyl, phosphate, sulfur. So then what is the difference? What makes fat so caloric? It is the level of reduction of the carbons that are part of the molecule. And I mean, you have this as part of your assignment, but you will realize that as part of biological molecules, as far as biological molecules go, the carbons that are part of the hydrocarbons of the fat molecules are the most reduced carbons that we can have in our body. The most reduced carbon in nature is methane, but we can't really use methane for energy. I mean, well, we can use it for energy, but not for biological energy. We can use it to burn it and generate energy. And as you know, methane is very, energy producing, if I can say that. So this is the most effective way to store energy. In addition to that, fat is also insoluble in water. It weighs less. It doesn't store water with it. Glycogen, for example, is not the same way. Because glycogen is hydrophilic, when it's stored, it also stores and packs water with it. And because of that, it's heavier than its own weight and adds weight by adding water to itself. And also because it doesn't mix with water, fat molecules usually don't affect cellular osmolarity. So it's perfect. You have a very efficient way to store energy that doesn't mix with water, it weighs less, and it doesn't affect osmolarity. It's a perfect way to store energy in the body. And in addition to that, it is also relatively inert. Now, it does contain energy. I mean, if you burn it, it's going to produce a lot of energy, but it's not reactive. So again, perfect storage, high energy, no osmolarity effect, weighs less, and it's non-reactive. It's a perfect way to store energy for the body long term. Fat as a molecule is also the primary source of energy for the liver and for the heart muscle. It supplies about 80% of the energy needs of the heart muscle and in the liver. And in resting muscles provides about 50% of the energy for resting muscles. And for hibernating animals and for migrating birds, it's virtually the source of energy for them. So fat is not an evil molecule. We should not be afraid of fat. We should be afraid when we have collected a lot of fat. But on the other hand, I like to joke with that by saying that we're just following the advice the church gives to have food storage. The only difference is that some of us are very diligent at following that advice, and we carry our food storage with us everywhere we go. And then I usually make a joke with some of the students who at your age are quite skinny. And I say, well, if we're in the middle of the desert, I will last a lot longer than any of the skinny people because I have a lot more deposits of fat and hence energy. So the likelihood of us being though in a desert, in the middle of the desert, is very low, but you never know. So for that reason, some of us carry our food storage, some of you barely have any reserves. So with that, let's move along. The objectives of these presentations are to understand the catabolism of fatty acids or beta oxidation. Now today we're not going to talk about beta oxidation. We're just going to touch on it. We're just going to define it at the end of this presentation. This is going to happen primarily next time. Next time we're going to talk about beta oxidation of saturated, even carbon number fatty acids. We're going to talk about unsaturated fatty acids, odd carbon number fatty acids. Then we'll talk about regulation, and one of my favorite topics, ketone bodies. Not because of ketone bodies are so great for the body, but because by understanding how ketone bodies work in the body, you actually will see one of the most beautiful cross pathways communication. That's why I enjoy talking about the ketone bodies, not because I would like for our bodies to have high concentration of ketone bodies for some reason. Anyway, we'll get to that. So when we talk about fatty acid catabolism, there are really three main entry points, I guess, or entry reasons that contribute to the blood concentration of fats or fatty acids. The blood gets its fatty acids and fats through ingested food, or by degradation from the body's deposits, or newly synthesized by the liver and transported. So these are the three main sources of lipids in the blood vessels, in the blood. So this diagram shows how fat is digested from food. Here we digest it, obviously. It gets into the stomach. Not much is done in the stomach. Stomach primarily deals with proteins and some carbs. There's mechanical mixing, but not much with the fats because there are no lipases. Also, fats are not very easily mixed with hydrophilic or aqueous solutions, so there's not much going on here. However, once it passes the duodenum, where the gallbladder excretes its bile salts and its content, basically, the bile salts act as emulsifiers. They make fat soluble, and the body now, or the intestines now, can mix fat with its aqueous environment and make it accessible for lipases that will be released from the pancreas, which is just right here behind the stomach. And the degradation will begin. We'll start generating single fatty acids, or diglycerides, triglycerides, monoglycerides, and eventually they will be all degraded to single fatty acids and glycerol. They will be uptaken by the mucosa and then packaged in triglycerides to form structures such as kilomicrons, and then the kilomicrons are now part of will enter the blood, and the blood vessels have these lipoprotein lipases that end up communicating with the apolipoprotein, that is this green part, that is part of the lipoprotein structures of the kilomicrons, and then these lipases degrade the triglycerides to single fatty acids and glycerol, and then they get transitioned through the blood vessel, enter cells, in this case shown myocyte or adipocytes, which are fat cells, where they get degraded or repackaged for whatever their purpose is. On this figure, you can see the structure of the kilomicrons, which are lipoproteins. We will, I think sometimes I get confused between 481 and 482, but I believe at some point we will talk about them again. I think we've talked about them in 481, or maybe we'll talk about them later in 482, but regardless, these are lipoproteins. It's basically a mixture of lipids and proteins, just like the name suggests. Okay, so that's one way that how lipids enter the blood, and from there it gets to the tissues. Another way is from the body's deposits. Hormones such as glucagon or epinephrine, acting through they're corresponding G protein coupled receptors. And how do I know that? Because they signal through GS, and also they're signal through adenylyl cyclase that converts ATP to cyclic AMP, which is a second messenger, things that we've learned in 481. And then cyclic AMP activates the cyclic AMP dependent kinase known as protein kinase A, or PKA. And once the protein kinase A, PKA is activated, then the kinase phosphorylates paralipins, which code the lipid droplets. Once they're phosphorylated, a different binding surface opens. At the same time, PKA phosphorylates the a lipase enzyme that is inside the cell, activating it. But at the same time, by activating it in these new open surfaces that are now available at the lipid droplet, they recruit the activated lipase that now gets in contact with the stored lipid droplet. These are, by the way, full of triglycerides. And then the lipase starts degrading triglycerides or triglycerols, maybe it's the same thing, to fatty acids, which are transported into the blood where they bind albumin. Again, fatty acids are poorly soluble in aqueous solutions. That's why they need a carrier protein, in this case albumin. Albumin gets them and transports them to their destination. In this particular case, this is shown here, myocyte. When it enters, it gets degraded to the beta oxidation to carbon dioxide, ATP, and electrons that are not shown here. So that's the second source of lipids for the bloodstream, and then for use by the body to be used by the body for energy. All right, now triglycerides are degraded to fatty acids and glycerol. So we're going to look at the how is glycerol and the fatty acids processed? How are they degraded? What's their fate in the cell? Glycerol represents about 5% of the energy carried by triglycerides. Now, to harvest the energy of glycerol, we need to use a known pathway. And, well, let me show you. Well, the question that I have is, which pathway is that? And what are the connecting metabolites? So let's start looking at what happens with glycerol. So glycerol, the first thing that happens to it is it gets phosphorylated with the use of ATP. And it's converted to glycerol 3-phosphate. Then glycerol 3-phosphate dehydrogenase oxidizes glycerol 3-phosphate to dihydroxyacetone phosphate. Now by now you should already be saying, aha, I know now how this will continue. And of course, triose phosphate isomerase converts dihydroxyacetone phosphate to glyceraldehyde 3-phosphate and then goes down the glycolysis pathway. So you can see that glycerol gets processed in a similar fashion as glucose. Now here's another question. Can there be another product as a result of this junction between these pathways? And by meaning these pathways, I mean the glycerol degradation pathway, which is basically these two enzymes, and then it becomes part of glycolysis, and glycolysis. These are the two pathways. Now, could there be any other product as a result of this junction of these two pathways? Is there anything else you can make from these, either one of these? And the answer is there is something else, and that something else is the starting metabolite of glycolysis, glucose. And we can make glucose from these by using the gluconeogenic pathway, and then by passing the conversion, the bypass basically of the first step, hexokinase, and the conversion of eventually getting to glucose from by following the steps of the gluconeogenesis. I didn't want it to start getting to each step because it's not the point right now, but it needs to go through the two bypasses, the PFK1 and the hexokinase bypasses, and it will eventually be converted to glucose. Now, this is a very important thing to understand, that glycerol can be used to make glucose by connecting it with gluconeogenesis pathway, and then eventually it will become glucose. The fatty acids represent 95% of the energy the triglycerols carry. Now, the fatty acyl-CoA that is in the cytosol can be used either for lipid synthesis, for membrane lipids, or it can enter the mitochondria and be degraded. But it doesn't happen right away, and it doesn't happen as easy as we may think. Now, in order for the fatty acid to enter the mitochondria, something needs to happen to it. Those that are short, 12 or below, free fatty acids, they can pass through the mitochondrial membrane. But those that are 14 or above, they cannot. And we need basically a transporter to get the fatty acid from the cytosol to the mitochondrial matrix. So how does that work? Well, there are three reactions involved in that transport of fatty acids from cytosol to the matrix, the mitochondrial matrix. The mechanism is known as the carnitine shuttle. The first reaction that needs to occur is to activate the fatty acid by converting the fatty acid to fatty acyl-CoA with the help of fatty acyl-CoA synthetase. Well, I think I answered my own question here. What would the addition of CoA do to the fatty acid molecule? Well, you know from previous examples that adding CoA activates molecules because it generates a high energy bond, meaning that it has enough energy to be used for work later on. So the difference between fatty acid and fatty acyl-CoA is that the fatty acyl-CoA is an activated fatty acid. Alright, so the conversion step of the fatty acid to fatty acyl-CoA are reversible, yet the reaction is not. I mean, look at it. These are reversible processes. We use ATP first to adenylate the fatty acid, and then basically to activate the molecule, and then CoA comes in and kicks out ATP. I mean, think about it. Why did we use ATP for? Simply to add a nucleotide to the fat so that we can kick it out. That's the whole purpose. And in the end, you end up with fatty acyl-CoA. These steps are reversible. Then how come the process is not? Well, because we're not paying close attention, and I'm trying to put your attention to look here and avoid this. Well, when you adenylate the fatty acid with a ATP, what's released is a pyrophosphate. Pyrophosphate is an excellent leaving group, but it's very unstable because the inorganic pyrophosphatase will degrade it to two inorganic phosphates. And that, as you know from the pathway of generating of UDP glucose, that reaction is highly exergonic and it's irreversible. And that is the reason why the activation of fatty acids is also irreversible. Even if these pathways are theoretically possible to be reversed, the absence of pyrophosphate makes this process irreversible. The energy of this of the fatty acyl-CoA bond is actually 31 kilojoules per mole, which again tells you that this molecule is activated. This high energy will be harvested at a later point. Alright, so that's the first reaction of the carnitine shuttle, to activate the fatty acid by converting it to fatty acyl-CoA. When the molecule is activated, the fatty acyl-CoA will come to at some point to the outer mitochondrial membrane, where with the help of the carnitine metabolite and the carnitine acyltransferase 1 enzyme, the hydroxyl group from the carnitine will basically bind at the carbonyl carbon, kick off CoA, and the acyl group now will be added to carnitine. And what we're going to have formed is fatty acyl carnitine. Once attached to the carnitine, the fatty acyl CoA is committed to oxidation. There's no going back. Once it's attached, it will be degraded. And now, then after this happens, usually happens in the outer mitochondrial membrane or the intermembranous space, the fatty acyl carnitine gets transported to a specific transporter that allows it to come through the inner mitochondrial membrane, because the inner mitochondrial membrane is extremely, how do I put it, sensitive in terms of what passes through. Almost nothing passes through unless it's specifically allowed by a transporter, or, well, by a transporter. There has to be a transporter. Nothing really goes through. Well, I mean, I kind of contradict myself right now, because I just told you that in this slide that those that are 12 or less can diffuse through, but you have to also understand this is a lipid membrane. Lipids would be able to pass through these membranes to an extent, of course. Now, once the fatty acyl carnitine is inside, there is an enzyme called carnitine acyltransferase, and with the help of CoA, we regenerate now fatty acyl CoA, and the carnitine goes back to the transporter, back to the outer mitochondrial membrane to accept another molecule of fatty acyl, so that it will bring it in as well. So that's the carnitine shuttle. Okay, so we have the fatty acids in the cytosol. They can be used for membrane lipid synthesis, or if they are bound by carnitine, they will be degraded in the mitochondrial matrix. All right, and the last slide I wanted to go over today is a simple overview of what beta oxidation is. Simply put, beta oxidation involves reactions with the ultimate goal to remove 2 carbons at a time from the fatty acid molecule. These 2 carbons will be in the form of acetyl-CoA. And the cycle starts at the carboxyl group. So basically, beta oxidation is cutting the hydrocarbon chain into 2 carbon segments that are bound to CoA, and the resulting acetyl-CoA now will enter the citric acid cycle to be burned to carbon dioxide and electrons. The electrons that are released during the beta oxidation are also loaded on electron carriers, and they are taken to the oxidative phosphorylation pathway in the mitochondria. And we'll see how that happens. So that's basically beta oxidation. Chopping the molecules into 2 carbon chains or units, and then activating it with acetyl-CoA for degradation. So here's a question for you that I'm not going to answer this time, but I'm going to let you answer it at home, and we're going to start from here next time. How many times the beta oxidation needs to repeat in order for the stearic acid to be degraded completely? How many cycles of beta oxidation a stearic acid needs to undergo in order to be completely degraded to acetyl-CoA? Alright, so think about it, and I will start from this slide, my next presentation. Thank you very much, and until our next time.